So to continue from our discussion on DNA from the last video and transition into our discussion of RNA in this one, we have the notion that we talked about 3 billion base pairs in a single human cell that constitute all of our DNA molecules. So that's 3 billion base pairs per cell. Now, that doesn't mean that they're all used to result in a protein. However, there are short stretches of these 3 billion base pairs called genes that will result in the coding for a protein or an enzyme. And all the genes of one person is called the genome. We have, it's been estimated, about 22 to 23,000 genes. The exact number is still not known. It's going to be a while before it actually is. Still lots of work to do. But it does turn out that this is only occupies an amount of real estate in a DNA molecule of about 2% or 1/50th. So out of those 3 billion base pairs, only 1/50th are actually meaningful in the production of proteins. The rest is considered junk. Now, that doesn't mean it's not needed or necessary, but that's a discussion for another course. So we have these short segments of the DNA molecule that are going to tell us how to build proteins. And our first step then is to transcribe these short stretches, transcribe them into RNA molecules. There are 3 types of RNA molecules. The first type is the messenger RNA molecule, or mRNA. These are your instruction codes for making the protein. So anytime you buy something new, right, you got to go through the instruction manual and figure out how to use it. The second major type of RNA is the ribosomal RNA. So the messenger RNA that's been made has to exit across the nuclear membrane and into the cytoplasm to interact with ribosomes that make up most of the ribosomal RNA that is found in the cytoplasm, either free or attached to the rough endoplasmic reticulum. The third type of RNA is transfer RNA. Transfer RNA supplies the parts. So let's go back here. So we've got the instruction manual from messenger RNA. We've got the manufacturing plant or place for ribosomal RNA, and all we needed was the part supplier, and that's transfer or tRNA. So now we're able to make a protein out of these components. There are 3 major differences between RNA and DNA. The first is that RNA is single-stranded. DNA, as you already know, forms a double-stranded helix. But because it's single-stranded, it doesn't have hydrogen bonds. And so the hydrogen bonds provide stability, recall, but RNA is not a stable molecule. In the lab, I had to work with it at 4 degrees centigrade. Just when I was working with it, I had to maintain as short a time as I could while I was working with it. And then when I wasn't using it, I had to store the RNA molecules in a minus 80 centigrade nitrogen-cooled refrigerator. So it's a pain in the butt to work with because of the lack of hydrogen bonds. The ribose sugar in RNA is the 5-carbon sugar as opposed to a deoxyribose sugar in DNA. So an oxygen is missing on the 5-carbon sugar group in DNA, but is present in the 5-carbon sugar group of RNA. And finally, RNA, recall, we already discussed this, has uracil as one of its 4 bases instead of thymine. So RNA will have guanine, cytosine, adenine, and uracil. So the essential function here for RNA is to take the code mandated in the DNA molecule, and after that DNA molecule is transcribed into the RNA molecule, then the RNA molecule's job is to exit the nucleus, go into the cytoplasm, and interact with the ribosomal and transfer RNAs in order to be translated into a protein. Now, how does it do this? Each of the 3 letters that make up the RNA molecule are called a codon. So as it turns out, every 3 letters in RNA is a word. So this is an extremely long book that has only 3-letter words like cat, dog, and, for, the. So only 3 letters make up this book. Imagine trying to write a book such as that. And as it turns out, these 3 letters will each correspond to a specific amino acid, because our job here is to build a protein, right? So we know proteins are made up of amino acids. And each of the 3 letters does so, and it's called a codon. Now, the relationship of a codon to the amino acid is governed by the genetic code. And the genetic code is the way we translate those 3-letter words into an amino acid. So there are 3-letter words that translate into one of the 20 amino acids. It turns out that the genetic code has 64 of these words. And so, as you might imagine, if there's 64 3-letter words, 20 amino acids, that some of the words point to the same amino acid. And this will become clearer when we look at the actual genetic code. Okay, so that, as we talked about, there are 64 possible 3-letter words. These 3-letter words called codons. 61 of them will actually point to an amino acid. There are 3 stop codons. We need some kind of guide as to when to stop producing the protein. We also need one of the codons to be a start, right? You got to have a start switch and an off switch. And those are handled also by the genetic code. So here is the genetic code. Don't be intimidated. You're not going to have to know it unless you take genetics, but you don't have to know it. And what you see here is the idea of the 3 base pairs that make up the 3-letter word that are going to correspond to an amino acid. For instance, if we look at the first column, second row, we see the letters LEU. And that is the amino acid. This is a shorthand for the amino acid leucine. It corresponds to these 4 3-letter words. In fact, if you look at the block above, you also see LEU there that corresponds to 2 more of them. So there are actually 6 codons that will correspond to the placement of a leucine at a particular point in a developing protein.